The Sucrase and Lactase Lab, you're going to be considering each of these solutions. So you're going to want to try lactose with sucrase. You've already done lactose with lactase, so you know how that works. You're going to do lactose with sucrase. You're going to do sucrose with sucrase. And then you're also going to do sucrose with lactase. The fourth one would have been the lactose with lactase, but like I say, since you've already tested that, you should know the result. Okay, so you're going to be deciding whether glucose would exist from these. So test tube, okay, so the test tube that has the lactose and sucrase, there would not be any glucose produced. In the sucrase and sucrose, you would have glucose produced. And in the sucrose with lactase, you would not have glucose produced. And of course, that fourth one, the lactose with the lactase, we would have glucose produced. So, which sugars are broken down by lactase, and how do you know? Which sugars are broken down by sucrase, and how do you know? Does the same enzyme digest lactose and sucrose? You should be able to answer those. Your results illustrate a general principle called enzyme specificity, which means that each enzyme acts on only a single substrate or several chemically similar substrates. This figure illustrates how even small differences in shape can determine whether substrate molecule can bind to the active site of an enzyme. This enzyme digests a substrate molecule in two product molecules. Which molecules can this enzyme digest? So look at the shapes. You should be able to decide which ones would be able to be digested by this enzyme based on the active site. And then explain your reasoning. Explain why sucrose and lactase are not digested by the same enzyme. So if you look at the structures of them, what do you notice? Because of enzyme specificity, your body needs lots of different enzymes to digest different types of food molecules. In the next section, you'll learn about another digestive enzyme. Okay, so here's where it's going to refer for you to watch the first 2 minutes of the video that is attached in Google Classroom. Experiment with salivary amylase enzyme. Near the beginning of the video, the teacher makes a mistake. He meant to say that starch in the presence of iodine produces the dark color purple, which is the same thing that we did. In contrast, when iodine interacts with short fragments of starch molecules, this produces a pink color. Explain why the contents of the beaker with saliva turned pink, but the contents of the other beaker stayed purple. And based on this experiment, describe how saliva contributes to the digestion of a starch-containing food like bread, tortillas, or pretzels. All right. If you have any questions on any of this, please let me know.